Okay, so here we have crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the cell division, the first one. AI. Describe the function of the S phase. This is where DNA replication occurs, okay, prior to mitosis / meiosis. Okay, that was easy. Now, some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. Crossing over is expressed in chromosomal regions near the centromere. So, near the centromere, we don't have any crossing over, essentially. And the reason is because it's a hypothesis, one or more of the kinetochore proteins in the centromere are responsible for the expressing of the crossing over around the centromere. Now, to investigate their hypothesis, the scientists modified chromosome 8 in yeast. So right now we're dealing with yeast, with chromosome 8 to be specific, so that if you have a pair of homologous chromosomes, which means you have 4 chromosomes, okay, some will contain the ability to emit red, and some will contain the ability to emit green. But this is before the crossing over. After the crossing over, one chromatid gives you the red, one gives you nothing, one gives you red plus green, and one gives you green alone. Okay. Double I. Explain why some haploid cells formed after meiosis in this experiment have only one fluorescent marker. So right now we're talking about the ones that are on the edges, the green one and the red one. Well, you should know that crossing over is only happening between the chromatids that are adjacent to each other from the different chromosomes. That means the chromatids at the edges do not undergo any crossing over. Okay, so we're going to say that these cells received chromatids that did not experience crossing over. So the chromatid either emits green or emits red. Okay. The scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence in a non-crossing over hotspot will affect the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore, which is this one, no kinetochore proteins. Group 2 contained the kinetochore protein CTF, and Group 3 contained the kinetochore protein IML. For each group, the scientists determined the frequency of crossing over between the RFP, which is the red, and the GFP, which is the green. Okay, to determine the frequency, the scientists added the number of the cells emitting both red and green light to the number of cells that emitted no light and divided by the total number of cells. Okay. BI, identify the control group for the scientist's first experiment. A control group is something that we are, let's say, it's missing the experimental part, right? So right now we're checking if we can, see the effect of individual kinetochore proteins being added. And so, for example, CTF is a kinetochore protein. That's not a control group. IML is a kinetochore protein. That's not a control group either. The control group is the one without any kinetochore protein. So we will call that the group called none, which, if you remember, that was called group 1. Or you could just say group 1. That's going to be our control, why it doesn't have any kinetochore proteins. BII. In a follow-up experiment, the scientists created a modified version of CTF in which the DNA binding portion, so its ability to bind to the chromosome, has been removed. It can no longer bind to the chromosome. They compared the frequency of crossing over in yeast cells in the presence and absence of unmodified CTF. So right now we have modified CTF. It cannot bind to the chromosome. We also have unmodified CTF, which is essentially this one here, okay? Which, as we can see, it has been able to bring down the frequency of crossing over compared to the control. This one decreases the frequency of crossing over, okay, by being able to bind to the DNA, to the chromosome. All right, in the follow-up experiment, justify why the scientists used the modified CTF that is unable to bind to the DNA as a control. Okay. Well, the best way to know the function of something is to remove that something. So right now the scientists are trying to find out if the CTF binding ability to the DNA does affect any crossover frequency. Okay. So by removing it, they can tell. So basically, to tell the function or the importance of the DNA binding ability of CTF on the frequency of crossing over. Okay, I. Identify the independent variable in the follow-up experiment. Well, the independent variable means you are controlling that thing. You can make it present or you can make it absent. And so, for example, here, the scientists were able to choose whether to have the presence or the absence of the unmodified CTF. So our control is going to be, our independent variable is going to be the presence and absence of unmodified CTF. Okay, C. Based on figure 2, describe the effect on the frequency of the crossover when CTF is attached to the chromosome 8 hotspot compared when the IML is attached to the hotspot. Well, I think we can tell that compared to the control, which is the none, CTF has actually brought the frequency of crossing over down, but compared to the control, the IML error bars are actually overlapping with the non-error bars. So it was not able to change the frequency of the crossing over. Okay, so now we're going to erase that and we're going to be writing that the CTF has significantly decreased crossing over compared to the non-group, while IML has no effect on crossing over compared to the non-group. Okay. So, D. Predict the effect on the number of the copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached. Well, we can tell here that CTF was able to bring the frequency of crossing over down. And so if you are basically CTF resulted in less crossing over, right? And we were told in the beginning of the question that crossing over is important for the separation of the chromosomes from each other. Okay, and so now if you affect crossing over and you bring it down, you will basically bring down or bring up the chance of non-disjunction. You're going to be increasing the chance of non-disjunction, which if happens during crossover mistake, this happens during meiosis 1. And if non-disjunction happens in meiosis 1, you can be expecting some bad results, right? Because what's going to happen is you will get 4 gametes. 2 of them will be N plus 1, which means they have one more chromosome above the normal, and 2 will be minus 1. They'll be having one chromosome below the normal. So how do we answer the predict part? We're going to say that the daughter cells will have one more chromosome or one less chromosome. Okay, because N plus 1 means one more chromosome and minus 1 means one less chromosome. And of course, we're going to have to justify that. We're going to say that CTF decreases crossing over frequency, so it can lead to more non-disjunction. Okay. All right. Triple I, explain how the presence of hotspots in Figure 1 could increase the likelihood that a population will survive in the presence of selective pressures. Well, let's go back to Figure 1 for a second. And in Figure 1, we have crossing over, we have hotspots, and we're told that the hotspot is basically the area of having a higher frequency of crossing over. Okay, well, if you have more frequency of crossing over because of the hotspot, you will expect more genetic diversity, which means that you will have more chance of survival and reproduction. So we're going to say it will lead to more crossover, so more genetic diversity, so more chance to respond to natural selection pressures, leads to some individuals surviving and reproducing more than other individuals. Okay.